Anyway, well, howdy. That's the term I guess really introduce people or introduce myself. Thanks for the introduction, Astrid. I'm glad to be here again and to give a seminar on some of the work I've done when I was at LSU doing my PhD and as a research associate. Can you hear me okay up in the back? Okay, good. So over the course of my career, I've worked with some interesting insects, and one of which was my work with fire ants, much of it which formed the basis of my PhD there. And so I'll talk a little bit about that, but I'm also going to talk a little bit about the work I've been doing with Texas Agrilife as a postdoc in Amarillo and what I'm currently doing in Weslaco and in collaboration with some members of your department like Creighton and Betsy on zebra chip disease. And so, so my intent was just to, hi Betsy. So my intent here today is just to introduce myself to you and just to give you an idea of some of the things I do and what I've done, and just sort of entertain you and just have some fun. And so I just wanted to give the disclaimer at the start here that there's going to be some disturbing images, but I think most of you here, we're all scientists, we can handle it. So, but anyway, I'm going to start off by talking about the red imported fire ant. Most of you, I'm sure, have encountered this insect. It's probably one of the most egregious pests ever to grace the soil of this country. This is a typical landscape in Louisiana and parts of eastern Texas where we have mound-forming fire ants. And they build these mounds, which in a lot of cases causes problems for agricultural machinery, and there's other issues, of course. Of course, the ant has a notorious reputation for attacking livestock and people. And so there's a certain segment of the population who have sensitivity to the stings of the fire ant. And on occasion we see very large populations forming in highly disturbed areas. And that seems to be their preferred habitat, is areas that are highly disturbed. Now, this insect is not a native ant. It was introduced. Oh, do you have a question? Disturbed are meaning that they've been cleared, they've been, they lack the ecology that would have normally occurred there. So if you had a forest and it was cleared, that would be considered disturbed. Disturbed also are roadsides where you have pastures or another highly disturbed area. Agriculture, lawns, pretty much anything that humans do to the landscape would be considered a disturbed area. So, and that, incidentally, fire ants do not do well in dense forests where there seems to be, they prefer open areas that, where they have free reign over the area. Where there's, as I mentioned here, there's no competition. In the ant world, there's a certain amount of competition going on between different ant species where none are predominant over the others. So they have a tendency to exploit resources or partition their resources in a way that no one species is dominant over the others. But in this case, we have a very depauperate ant fauna in the southeastern United States because of the massive land clearing that has happened. So we don't have the original ant fauna that was here before the fire ant was introduced. So what happens in a lot of instances when you introduce an exotic organism, its natural enemies that had kept it under control, usually predators or parasitoids or competition, these were lacking when this insect was introduced. And this enabled it to rapidly expand over the landscape. Here's a current map showing the distribution of this ant through the southern United States. And it was introduced to this location here, Mobile, Alabama. And through a course of, the famous eradication program of the USDA in the 1950s and other, it was, I guess now in hindsight, was rather ill-conceived and poorly implemented and really probably enhanced the spread of this insect. Now it occurs in Puerto Rico and California, Arizona, China, and Taiwan, and Australia as well. So it's showed up in a number of areas and continues to spread into other countries. So because of that, because of the failure of chemical management, there was an effort to look at, okay, well, if there are no natural enemies here in the United States, are there natural enemies in Brazil, where this ant originated from? And it was discovered there were a number of insects known as decapitating flies. They call them the genus Pseudactyon. And there's a complex of some 20 or so different species that partition the ant host itself as a resource. So different species of decapitating flies will attack. They have their own preference for a different sized ant. So there's those that, for example, this one here will attack smaller ants, the smaller workers, whereas this one here would attack larger ones. So they partition their resource that way by size. But also they partition it by time of day. So there are those flies that will attack their ants either in the early morning or afternoon or some other time of day, or even year. There's a seasonal partition as well. And so this enables there to be a large complex of insects or parasitoids that can attack this ant. Now, the effect that these flies have on the ant is quite interesting. It's not really the amount of mortality that these flies exert on ants, but rather their effect on ant behavior. These ants, well, they have to feed their colonies in order to grow. So they go out and they forage, they look for food, and they alert their nest mates, and they recruit them to resources. So they all come out and try to obtain that resource before other ants discover it. And what often happens is neighboring fire ant colonies will go to war with one another. They'll fight over these resources. And this releases chemical compounds in the environment, which attracts these insects to them. So these flies can actually smell their ants and zero in on them and attack. You can see there that these flies are extremely tiny. Now, despite the name decapitating fly, it doesn't mean that they actually go in and bite their heads off and you know, like this. It doesn't do anything like that. But rather, it's the behavioral effect. Now, you can see here, there's flies that are attacking ants. Here's one here is about to see, it just got parasitized. And these flies will, in a fraction of a second, will dive in and inject an egg into the ant's thorax, the middle part, for those who haven't had your entomology classes. And so you see that these ants will race around. You know, if they've been stung, they'll run around, try to attack the fly and catch it. And so when this happens in the environment, ants will actually abandon their resource and go back to the nest and say, "The heck with it! I'm not going to bother this. We're under attack. So let's get out of here." And you know. And this is what happens. So in a sense, the colony is less able to feed themselves. And so the populations, theoretically, should decline. Yes. Anyway, so we all have our, you know, we've all probably seen some zombie movies in Hollywood and see the uncanny resemblance here. So, but, you know, we have this notion in our minds of what zombies are. It's something that takes control or attacks another. It seems to have no control over itself. Like a virus or something has taken over it. Incidentally, I was meeting with my colleagues at the Brackenridge Laboratory in Austin, where they are also doing work on fire ants. And it was our lab at LSU that had discovered this zombie ant phenomenon, and they've sort of taken off with it, and they're continuing this research on zombie ants. And I was told that there's some interest in Hollywood to make a movie about decapitating flies attacking people. And so I thought, well, that's pretty cool, but maybe they'll funnel some of the profits my way. But anyway, there's also zombies in the insect world as well, where these parasites, whether they be nematodes or virus or fungi, will induce a change in behavior in their host that will benefit the survival of the parasite. So in the case of these nematodes that infest crickets, the crickets will search for a body of water and drown themselves so that the nematode can emerge in a suitable environment. With fungi, the affected insect will climb up to a heightened location where the spores can disperse efficiently. And in the case of a virus, this nuclear polyhedrotic virus, where caterpillars will also climb up to the upper foliage, and where the virus, the hemolymph, the blood, will drip out of the insect and land on the leaves below, where other caterpillars can consume it. And so it's this kind of zombie behavior with this host-parasitoid association, where the parasitoid fitness depends on what the host does. So the host must not die, at least not while the parasite is inside of it, because its survival relies on what the host is doing. So it has to alter the host behavior and manipulate it so that it can survive at the expense of the host. Now, in the case of the fire ant, their normal course of activity is to go out and forage for food. After these insects, or ants rather, have been parasitized by a decapitating fly, they stop that behavior. They go back to the colony and they never leave again for 2 weeks. And this is something that we had tested in the lab, where we exposed ants to heavy fly predation for hours or days, so we had a high level of parasitism, at least we hoped to. And then we followed the activity over a period of weeks. What was the fate of these ants after they were parasitized? And what we found that after about 10 days after they had been parasitized, they would start to mass at the ends of these artificial foraging tunnels. Of course, we wanted to be able to see what they were up to, so we had to use clear PVC. But what would happen is these zombies would start to mass at the entrance of these tunnels, and then just a whole flush of them would start emerging, and then walking around the arena for several hours, looking for a place where they would eventually die. So presumably what's happening here is the parasitoid well, I'll explain here in the next slide. The life cycle of this parasite is, well, the egg is injected into the thorax, the egg hatches, and then the maggot migrates to the head of the ant, where it develops within the ant's head. Okay, it consumes all of the tissues during this 10 to 14 day period, except the brain. It leaves the brain till the very end, because it needs the brain for when it's searching for a suitable place to pupate. And it pupates inside of the ant's head. So what's happening here is, this is the maggot here. It seems to be what we think is secreting an enzyme, the biochemistry that we don't understand, what the biochemical basis is here. But it's secreting some chemical that's inducing a cascade of behaviors where the ants will, again, it'll enter these entrance tunnels, exit them, and then start wandering around the environment, looking for a suitable place for the maggot to pupate, and eventually it decapitates the ant. So what it means by decapitating, it secretes an enzyme that breaks down the soft tissues of the legs and the head, and eventually they just fall off. So what is happening is these flies appear to be using the ant as a vehicle to locate a suitable place to pupate. And this is usually a dark, moist place, so this is something where the fly pupa would be spared desiccation and other nasty things. So going on from there, we wanted to look at, okay, we were introducing these parasitoids to the U.S. for biological control. But we wanted to understand, okay, when we're releasing these insects at a point in the area, we want to know how fast they would be spreading over time so we could predict where they would be at some future event. And so there's a couple of theoretical models that describe spread of population over time, whether the they spread at a constant rate, or they're just moving at a constant speed over time, or whether there is a phenomenon known as jump dispersal, which I'll explain in a moment. But depending on what pattern of spread they have, it depends, or it will tell us how fast they will spread. One, if they have a constant rate of spread, here in this example, they've spread maybe 15 increments after 10 time increments, whereas here they would spread 10 times as fast with this other type of dispersal. And so we looked at this rate of dispersal, how these flies dispersed across the landscape over a period of years. And for example, here we mapped out where the flies were located during their initial expansion in this place. This is north of New Orleans. And we went out from there. So we had 2 releases, one here and one about here. It's north of Baton Rouge. And so we followed by searching for these flies in the environment, where they were and where they weren't. We were able to determine approximately how fast they were moving. And so you see over this time scale just how fast these insects were spreading across the landscape. And so we took this information and derived a model to tell us, okay, what pattern of expansion did it have? Well, clearly it's not a linear rate of spread, but rather this quadratic, which is characteristic of something called jump dispersal. So these flies were racing across the landscape, and so we understood something about how these insects. Okay, so if they're spreading this fast, where does this mean we need to do our releases over a spatial scale? So if we don't want them too close together and we don't want them too far apart. We want to have just the right kind of overlap so that they'll quickly merge in the shortest possible time. So jump dispersal is, well, let me explain here. This is where you have your initial population here, and in that first time scale, they've spread just that little bit there. But over time, as this original population is spreading, there are always a few individuals that spread just a little bit farther than the others. And they start their own little populations well ahead of the advancing front. And so this is what they call jump dispersal. And you look a little further down, okay, well, still more little populations are forming way ahead of this original population here. So we have the central population that's spreading on its own and coalescing with all the other ones that are in turn forming and spreading and in turn. And so this results in really quick population spread over space. We were also interested in, okay, well, what are the flies doing during the day? Presumably they have an activity rhythm just like we do. And so we just, just to show, okay, when are the flies most active during the day? And so, well, they're pretty much not active early in the morning and late in the afternoon, but in the middle of the day is when they're most active. So, but also seasonally, when are they active during the season, and what environmental variable can we use to predict what their population sizes would be? So we looked at lots of things like soil moisture and temperature and all kinds of things. We just want to know what could we use to predict the population sizes. And we discovered that soil moisture levels were very well correlated with the populations of the flies. And so that has some usefulness, I think, in terms of modeling based on the soil moisture levels. Now, going away from ants now, look at some other insects that I worked on at LSU. The mole crickets, I'm sure we've probably seen those here as well. These are another invasive insects from Brazil. And of course, people who like to golf and all that, they hate these things. And they're a turf grass pest. And, well, of course, mole crickets sing. The males sing, they stridulate, they sing, and they attract females. And we use that behavior to sample their populations by using these artificial cricket traps. So there's just a battery-operated speaker that plays the same frequency that the mole crickets do so that we can trap them. But there's also a parasite. It's called a red-eyed fly. It's also a parasite a parasitoid of the mole crickets. And these flies also have ears. And so they can actually hear the songs of the crickets and find their hosts that way. So it's kind of a peculiar association there. There's another invasive weed, Salvinia. This is a big problem in waterways in the southern U.S. It's another invasive from Brazil. And so we've imported a number of weevils that are predators of Salvinia. And let's see. Here's a series of pictures showing the effect that these weevils have. Very quickly, in a span of a little over a year, these weevils will effectively clear a lake or body of water of this plant. So that's pretty interesting. Now, getting to the work that I was currently doing with my postdoc and currently in Weslaco with zebra chip disease. Many of you are probably familiar with this disease and its relation to citrus huanglongbing. Both involve psyllids, not the same species though, but they both involve psyllid vectors and a similar pathogens, Liberibacter. And so we've been trying to understand what the basis is for this, the pathogen, how it affects the plants, and how it affects the vector, and how it transmits the disease. For those of you who are not familiar with zebra chip disease, this is some of the symptoms that are commonly observed in the field. Leaf curling, shortened internodes, purpling, leaf scorching, aerial tubers, and just early plant death. So this is a lethal pathogen. It's a phloem-limited bacteria, and the impact, though, is quite severe on the tubers. It causes the discoloration. This is where they got the name zebra chip from. Some alterations in the physiology of the plant, which apparently changes the way the sugars and starches are allocated within the plant, but also there's necrosis, and so we have a high level of reducing sugars in the tubers, which, when cut and fried, results in this kind of issue. So obviously this is unacceptable for those who enjoy potato chips, but also other potato varieties. All potato varieties are affected by this disease. So some of the work we did up in Amarillo involved, trying to understand what the distribution of the disease was in the environment. You know, what is the spatial pattern of the disease? We often see these clusters of diseased plants, so we've looked at how these plants, the disease evolves over time, as shown here. So we have, disease progress curves, and so we have some knowledge of how the disease evolves and progresses in commercial potato fields. Now, my interest is in vector management and how we can best control the potato psyllid and keep them, from affecting the crop. So one of the things we're doing here is evaluating these insecticide-treated barriers. Now this is fabric that's treated with pyrethroid, permethrin, and this is a product that's used extensively in third world countries for malaria control, or controlling the mosquitoes. So people put this stuff in their doorways, over their beds, and so when a mosquito encounters this, it dies. But so the thought is, well, could we use this for crop protection as well? And obviously this wouldn't be practical on a large scale, like 100 acres or so, but in Mexico and other Latin American countries where potato growers commonly grow them on a smaller scale like this could be of some value. And so we're just now we're harvesting the plants from these plots and collecting the data. So I can't really say whether this is working or not. It appears to be. Of course, we're evaluating new insecticide chemistries to, as old products are phased out or removed from the market, new ones are coming in, and we need to evaluate them for potato psyllid management. Looking at also integrated pest management and incorporating different kinds of organisms and chemicals and evaluating them at a large scale like this. And looking at evaluating different varieties for resistance to the disease. And this is work done here with Creighton and Betsy and Doug, where we have these cage trials where they've planted different varieties of potatoes, and we've introduced psyllids carrying the pathogen and determining whether these different varieties have different levels of resistance. And I'll let Creighton and Betsy talk about that at some point. We're still working on this, but this is this whole plant breeding is really important in terms of trying to develop resistant varieties to the insect and the pathogen. We've also been looking at the role of this pathogen within the insect and how it affects the insect and how the environment affects the pathogen within the insect. And so we were doing some molecular biology, looking at the titer levels of the pathogen within the insect at different temperatures and how different temperatures affect the titer. So we have the highest titer in the range of around 20 to 27 degrees Celsius, which is also the preferred temperature for the insect. So they both do very well at that temperature. But how does the titer change within the insect as it ages or as it changes from a nymph to an adult? And we routinely see very low titer levels in the nymphs, but as they molt into the adult and age, the titer levels start to go way up. But by sex, though, there's no difference between males and females with the titer levels. So in some insects there are. Different vectors have different, the sexes have different titer levels. But also, okay, well, you're looking at your different colonies. When you're maintaining psyllid colonies in your lab, and you're wanting to use these insects for experiments, we want to know, well, how many are in your colony are carrying the pathogen? Is it a constant level or does it fluctuate? Well, I think the answer is clear there. In fact, in some colonies as we sample them over time, the numbers of individuals that are positive for the pathogen will decline to 0 and then start to reappear again. So we don't understand what's going on there yet.
Does it take into account population structure? So, for example, if
it could.
Nymphs, then you would expect the population to crash?
We don't know. You know how, like it's not like we're dealing with a large organism like a cow or something where you can easily observe and mark every one of them and follow them. But these are so minuscule that we just can't follow them. But yeah, even during the day, Betsy, we'll see fluctuations in the average titer levels. So if you're using your insects at this point in time versus this point in time, you have very different titer levels. So their bacterial populations within the colonies are changing through the course of the day. And so, well, let's get to the fun stuff here and discuss really just some of the most, well, horrifying or disturbing insects that some people have the misfortune of encountering in their daily lives. And one of these is the Japanese giant hornet. Apparently, some 50 people a year die from this insect in Japan. It's like they don't have enough problems already. They have these giant hornets. And these hornets are notorious for wiping out honeybee hives. I've heard that 30 of these will decimate a colony of 30,000 honeybees. Just dispatch them in no time at all. And here's a video that describes
with severed bee heads and bodies. A few remaining soldiers make their last stand at the entrance.
Hold on.
Nearly every bee in the colony is dead or dying. Now, the hornets will take what they came for. They will gorge themselves on honey, but their chief plunder is the honeybee larvae and pupae that can feed their young for weeks. Then, as quickly as it began, the slaughter is over, and all is quiet again.
Now, these are European honeybees we're talking about here that have no defense to this. Now, apparently there's a Japanese honeybee where they are not easily dispatched like this. So they'll actually form a cluster, a ball, around these hornets and will raise the temperature so high that they cook the hornet inside the ball. And so they're able to, they've adapted to dealing with this kind of predation. So it's pretty interesting. Here's another insect that is really one I wouldn't want to encounter personally. The bulldog ant, native to Tasmania and Australia. Very primitive ant. Evidently, it has the lowest chromosome number of any animal. Its entire genome is on one pair of chromosomes. And of course, that's the females. The males have only one chromosome. So, but these ants they have very highly developed vision, and they could see you from about 1 meter away. And if you piss them off, they will leap at you, and that's the name bulldog ant. Now, this one is by far a very vicious animal. The bullet ant, native to really the tropics of Central and South America. I don't know if any of you have encountered this insect or not. Betsy, you have? You been stung by one of these?
No, but it sure cures your desire to eat against the tree.
I've heard that they actually will forage in the trees and they're known to shriek or something like that before they come at you. I don't know if that's true or not. But there's this famous index called the Schmidt pain index, developed by an entomologist working at the Carl Hayden Bee Lab in Arizona, Justin O. Schmidt, developed this pain index, and he claims to have been stung by the majority of these insects. And so we have a range of pain indices, and a lot of it, the descriptions are like a wine tasting or something. Like, for example, the sweat bee. Light, ephemeral, almost fruity. A tiny spark has singed a single hair on your arm. So that gives you an idea of what the sting of the sweat bee feels like. Okay, well, fire ants. Have most of you been stung by fire ants? Okay, well, look how low it is on the pain scale. Okay, red harvester ants I just was recently stung by a red harvester ant down in the Rio Grande Valley. And let me tell you, it's, something like using a drill to excavate your ingrown toenail. That was not pleasant at all. Bold and unrelenting, yes. Now, the bullet ant is off the scale, okay? This is pure, intense, brilliant pain, okay? The description of being stung by a bullet ant. Now, here's a video.
Meet the bullet ant. Its sting is said to be the most painful on Earth. Some liken it to being shot with a bullet. But you're about to see people stick their hands into gloves swarming with these nasty creatures. Among the Satere Moe tribe of Brazil, it's how boys become men. And for these young initiates, the painful moment is at hand. The ants have been gathered from the forest, then drugged, then placed, stinger first, into woven glove liners. Now, as the ants writhe in angry desperation, the ravaging begins. One by one, the young men suffer the shock of having dozens of stingers penetrate their flesh. They must wear the gloves for 10 minutes. Their agony is unmistakable.
The pain is incomparable, understand? There's no pain like it.
Have you ever had your hands on fire? It's the same thing.
And this moment's not over. Once the gloves are off, the stinging and burning only grow more excruciating. As the venom turns their hands into swollen, simmering, paralyzed stumps. What's worse, no initiate can be considered a true warrior until he has undergone this rite of pain, not once, but 20 times.
Yes, I don't think I could do it myself. Anyway, well, I want to thank you for your attention. I hope you enjoyed it, and it was a pleasure meeting you all. And if you have any questions, I'd be happy to answer them. Thank you.